You may have heard if anybody listened to the State of the Union address in January, President Obama talked about frontiers in the human brain to develop new innovative technologies and initiatives around the human brain, and that led to a flurry of discussions at the NIH, at other sister organizations such as DARPA, the NSF, to think about what the federal government can actually do to think about more about the mechanisms of brain function. And this is really critically important when you think about an aging population with burgeoning Alzheimer's disease, Parkinson's disease, and certainly for NIDA, substance use disorder. It is a brain disease, and we need to think about what are the underlying basic mechanisms of the brain. So based on the President's remarks, and then later in April, a press conference at the White House launched the BRAIN Initiative that will be about $40 million from NIH to launch this program, and $140 million overall when you think about all the agencies that will be involved. A steering committee has been put into place to think about the initiatives that will be implemented in fiscal year 14. It's being chaired by Corey Bargman at Rockefeller and Bill Newsom at Stanford, and the meetings are happening every month to think about initiatives. There's a meeting coming up in New York at the Rockefeller Institute to think about where we're going to go in fiscal year 14 and thinking about how do we really take on this very daunting and challenging task of thinking about the brain and all it can do just in terms of normative processes, but also implications for diseases of the brain. So a very interesting program that's being developed over the next several months, and hopefully you'll keep an eye out for that. The other thing that's going on is big data. Big data is everywhere. Just on NPR, I think yesterday I heard an interview about big data and Google, and we're hearing about the recent issues with phone numbers and the National Security Agency tapping into huge amounts of phone records and internet data. But it's also hitting the biomedical community, and for NIDA in particular, when you think about brain imaging data, it's a huge amount of information that's captured in brain imaging, and we're collecting data much faster than we know what to do with it. The Genome Project has given us some hope that there is a way to tackle it. There's sort of the geneticists are out ahead of this, but NIH has taken this on in a big way by establishing a program that will start in fiscal year 14 to put at least $100 million into these four areas to facilitate broad use of biomedical big data, develop analysis methods and software, importantly enhance training, particularly for early career investigators who are now starting to take hold of this information age and this wave of data that's coming out in the biomedical community, to give them the tools they need to analyze these data, from electronic health records to electrophysiological data to brain imaging and beyond. And an important aspect of this will be to establish centers of excellence where these sort of new tools and new technologies can be developed and disseminated broadly to the biomedical research community. So this is a very large enterprise being undertaken under the NIH Office of the Director. Dr. Francis Collins coined the term BD2K, so I can't take any responsibility for that. But it's really an important area when thinking about the future of biomedical research. We just need to get a handle on large data sets, the ability to share, and the ability to analyze these massive amounts of data. The other thing that's going on through the Office of the Director is enhancing diversity training experience of the biomedical research force. It's not the case, as it used to be, that everyone that goes through Ph.D. and postdoctoral training ends up in a research career, at least in an academic research career. There are many more opportunities, as you know, to use your skills and abilities in other areas of science, from government to industry to other venues. And NIH has taken on the role of increasing the idea of giving these training opportunities early in the career of pre- and postdoctoral fellows, so they can think broadly about the future of their career, not just to be like your mentor to do academic research, which of course we hope many of you will do. NIH, after all, is a research funding organization, so many of the people that we do support in training do go on to become independent academic researchers, but we're just realizing that there are many more opportunities. And programs like this, the BEST program, are designed to give students early on in their career an opportunity to think more broadly about where they might go in their research, their careers in the future. So I think it's a really good keeping up with sort of where we're heading as a society and thinking about where science can really make an impact. It's just not in the lab, but in many places in our society. And I think this is a good program to adopt. The other aspect of diversity, and it came up in Dr. Carlisle's talk, is diversity in our workforce, our ethnic minority diversity. And unfortunately, really over the last 30 or so years, NIH has not done a very good job of increasing diversity in our workforce. If you look at the 2008 census of the United States and compare it to faculty at a full-time medical school, or even worse, NIH-funded principal investigators, we have a lot of work to do. And it's been recognized recently by Dr. Collins himself and the Principal Deputy Director of NIH, Dr. Larry Tabak, and they have taken this on as a personal mission to improve the diversity of the workforce. And it makes so much sense when you think about creativity and bringing in new ideas. We need the diversity in our research force. We need people from different backgrounds, different perspectives, to give us that perspective that otherwise we would lose. So it's really a critical, important area to address. And based on many discussions for the Advisory Council of the Director with Dr. Collins, a plan has been put in place to launch the Building Infrastructure Leading to Diversity program. And I think this is really important to what Drew and the mission to enhance the research experience of undergraduates. I understand that's an important collaboration that goes on with Drew and local universities. Also to establish a national research mentoring network. We're finding that a lot of times ethnic and minority individuals don't hook up with the appropriate mentor. They lag behind in terms of the training, and this is an opportunity to make the mentorship more available and more easily accessible. It's been shown that you've probably seen the reports in Science about fairness and peer review. There's attempts to do better there as well. And importantly, a new chief of diversity will be named at the NIH to work directly out of the NIH Office of the Director to coordinate these efforts moving forward. So again, a major push to really bring in diversity into the biomedical workforce at the NIH. I think it's important and frankly long overdue. The other thing that's happened just in the last 3 years is that the National Center on Minority Health and Health Disparities has become an institute, and that gives it much more clout at the NIH to do a lot more and bring in a lot more programs that I've listed here. And I went online and I found that I think the research centers in minority institutions, Charles Drew actually has a center here under NIMHD, so I think that's really great that you're taking advantage of these kinds of opportunities. The NIMHD will also be the lead agency at NIH for addressing issues around the Affordable Care Act and health disparities. So they're playing a major role in bringing those issues to bear at the NIH. So a very important institute that's been raised to a higher level from becoming a center to an institute. And we're hoping that more institutes will partner with them and work on issues around health disparities and moving forward. NIDA has done a lot already and done so for many years in this area. In addition to being the acting deputy director of NIDA, I'm also the acting director of the Special Populations Office at NIDA, as if I don't have enough to do, but I really have taken this on as a labor of love, and it's really been quite heartening just to sort of take over a program that's been very well established in providing diversity supplements for ethnic and minority students. The summer research with NIDA, we give an opportunity of 70 high school students to work around the country in various labs, and I think maybe UCLA may be involved in some respects there in helping bring in high school students to get that first experience. We know that's so important in energizing students and kids to think about science as a career. It's really been a vital program. And of course you know about the DIDAR program, given Dr. Friedman's support of that program here for many years, and we think that's a great opportunity, again, to reach out to diverse populations, to give that experience and training that's so needed. And in our intramural program, we've been very successful in bringing in diversity as well. And there are some very important issues that arise when you think about drug addiction and health disparities. And I've just listed a few here, key issues that we are addressing at NIDA, from understanding the pattern of substance use in ethnic and minority individuals, understanding the potential differences in biology and risk and protective factors. Stress is a key issue that may in disproportionately affect underrepresented minority individuals, and that could lead to substance use and other disorders. So many areas where we're trying to reach out to and address the issue of health disparities when it comes to substance use disorder. But most importantly, all these things I've talked about, and this comes from the Office of the Director at the NIH, is to really reinvigorate the research community by providing many opportunities for funding, whether it's through the Pioneer Award, Eureka Award, which is for innovative research, the K99 R00 Pathway to Independence, which I'd be happy to talk to any of you who are in the postdoctoral arena, stage of your careers. It's a very good mechanism to get you from postdoc to independent scientist very rapidly. We have a number of early career mechanisms, the Behavioral Science Track Award for Rapid Transition for newly independent behavioral scientists, Imaging Science Track Award for newly independent imaging researchers. A-START is for newly independent AIDS researchers. I'll highlight the EChem. That's for early stage medicinal chemists. It's vital to what NIDA does in thinking about developing medications for treating substance abuse. We need a strong cadre of medicinal chemists, and unfortunately the average age of a medicinal chemist right now, I think, is about 75 years old. So we're having a hard time. We want to attract younger individuals into this area, and we've come up with the EChem Award to make it more attractive. Medicinal chemists are pulled into industry, as you might imagine, where salaries are quite good, and et cetera. But we're trying to do something because our medicinal chemists aren't going to be around, at least not working much longer, for sure. And of course, we have the diversity programs, which I've already mentioned. But I think the take-home message here is that because NIH, when you think about the NIH, you think of it as a monolith, the big black box. You put your grant in over here. 9 months later, if you're lucky, money comes out over here. It's like giving birth, it takes that long, unfortunately. That's not my fault. Yeah, if you're successful. So, there's a lot of moving pieces. There's study section, there's advisory councils, there's different funding mechanisms that I just shared a little bit of the sort of the tip of the iceberg with you. So I think the important thing is you need to reach out to a program official. I think any of the more senior level people in this room, like Henry or Chris, would say that knowing your program official is probably a good thing in terms of navigating the NIH and NIDA in particular. So if there's one thing, if the only thing you remember from my talk is this slide, you're doing well. And this is the reason why you should talk to program staff. Again, I talked about the funding mechanisms, program priorities. Where is, given particularly with limited budgets, where are the hot areas for NIDA? Not that you should always just follow the money, but it might be good to know where some of those harder areas are relative to things that are less emphasized. And for all the other reasons here, it's good to contact NIDA staff. So that's sort of a quick whirlwind tour of the NIH. I hope I still have you all. So I'm going to move on to some highlights of NIDA research, and I've sort of tried to go very broad because, again, I said we go from molecular biology, cellular biology to criminal justice cells, so there's a lot to cover, and I'll try to touch on a little bit of everything. And just so to give you sort of perspective of where we are, NIDA is a research organization at the NIH, and its major mission is to support and conduct research across a broad range of disciplines, as I've mentioned. But I think the important thing is the science must be useful and used. So we're just not here to talk to ourselves. We really want this information on what we know about the addiction as a brain disease, et cetera, to get to policymakers, treatment providers, and people interested in preventing substance use disorder. It's not just about the research only. Well, that's a major component, of course. We need that strong foundation so that we can translate that to effective prevention and treatment strategies. And I've alluded to the budget issues that we've had. You can see that in 2012 we were about $1 billion in funding for NIDA. With the sequestration now official, we've lost about $60 million in funding this year. As I mentioned, we've been able to do sort of a softer landing than we thought, because what we've been doing, not to get too down in the weeds, we've been funding a lot of 2-year grants that turn over very quickly. so that we were able to maintain less of a commitment base than otherwise if we had funded 5-year projects. So our turnover has been more rapid. That has allowed us to fund, that will allow us to fund almost as many research project grants, the R01, R03, R21, as we did in 2012, about 40 fewer in 2013 relative to 2012. Of course, if this continues, that will not continue to be the case. And as I mentioned earlier, our clinical research, our clinical trial network, for example, we're not funding the larger, bigger million-dollar projects on developing new treatments, testing new medications that we would otherwise have done had we received the full amount of funding. And the projection, the president's budget on the far right, we're hoping to get back up to where we were before the sequestration. Who knows? You know, it's politics, it's Washington, anything goes, right, as you've probably been following. So we'll see what happens. But just to get sort of to where the heart of the talk, of where I want to just focus, is thinking about addiction and what it really is. And it's a complex disorder, as we know. As Alan Leshner used to say, when he used to go to cocktail parties, everybody said they knew the answer to addiction. You know, they had a relative who quit right away. We had Nancy Reagan, just say no, or the fried egg, you know, this is your brain on drugs. Unfortunately, it's much more complicated, and we really think about it as a disease that's both genetic in origin to some degree, involves experience, and it really has a developmental aspect to it, a critical developmental period that I'll talk about. So to really, when we're thinking about just understanding addiction, as we need to think about all these factors. And importantly, we have to realize that not everyone who takes a drug becomes addicted, and this is where these individual factors really play out. And so we've come up with this sort of cute name, Gene Environment Development Interaction, or JEDI as we call it at NIDA, not to be confused with JEDI. And this is the inside joke, because when we thought about the acronym, everything in government is an acronym, our Institute Director, Nora Volkoff, had never seen Star Wars. So the first thing we did was to purchase the DVD so she could at least get the joke, you know, why the Jedi and Jedi. She never knew about Obi-Wan. But anyways, now she gets it. But anyway, why do we think there's a genetic component? Well, early on in NIDA funding, we had many twin studies that really showed a huge heritability component among our twin cohorts in substance use disorder. And from that original finding, we're now, with the technology in genetic technology and sequencing and doing genome-wide association studies, we're able to go further and say, okay, if there is a heritability aspect to addiction, what is it? What are the genes that are actually involved? What are they doing? What makes them functional? What makes that an important part of the addiction problem? And at NIDA, we've sort of taken on this approach where we go from looking at human genetic studies involving genome-wide association study, deep sequencing, and other methods to look at the genes that are associated with. Now remember, just associated with being addicted, we have no idea what these genes do. Take those genes and bring them into an animal model, whether it's using transgenic mice, or now even the rats we're starting to be able to do this in rat models and other ways. Look at the animal genetics to see what they actually functionally do in the brain and other areas of the body, and then bring that back into thinking more about the human condition of addiction. So that's been really quite rewarding, and the biggest finding really to date has been around smoking and nicotine, and I'm heartened to see that much of the conference today is focused on nicotine. And what's come up is there's a haplotype on chromosome 15 that came out as very strong, a single nucleotide polymorphism, specifically on the alpha-5 subunit of the nicotinic receptor. And for those of you who don't work in this area, it sounds like a bunch of gobbledygook, right? What am I talking about? But if you think about the nicotinic receptor, it comes in two flavors, an alpha flavor and a beta flavor. In a pentameric structure, 5 units come together to make the pore, the circuit. Henry, I think, and Theodore will talk more about that later today. But what we've been mostly focused on, we think about the medication that's most effective now, Chantix. It really targets the alpha-4 beta-2 subunits, which are the most prevalent in the brain and the body. But what this genetic study showed us, which is really quite intriguing, it isn't that those receptors at all that are relevant to, at least to smoking addiction based on these studies. It's the alpha-5 and alpha-3 and alpha 3 and beta 4 in that cluster on chromosome 15. But what makes the story even more interesting is that, remember I said we can go back into the animal and start to look at what are these receptors or genes doing in a model system. We're able to pinpoint that alpha 5 subunit, and it's in a very restricted area of the brain known as the habenula, which is involved in affect and emotion and actually negative affect. And there's a projection from the habenula where the alpha 5 is also found in the interpeduncular nucleus. And this pathway then also projects to the ventral tegmental area, the ventral striatum, and that's the holy grail of substance abuse. It's where the motivated behavior for substance abuse really happens, is this ventral striatum, ventral tegmental area. So we're thinking that this loop from the medial habenula to the interpeduncular nucleus is impacting on the dopamine pathway to actually put the brakes on the ventral striatum. And in persons with polymorphisms to the alpha 5 subunit, that alpha 5 is not working so well. So that this pathway is not operating optimally as it would in people without this particular variant. And what you can do then in the mouse, you can mimic that suboptimal gene in the human by knocking it out, literally taking it out of the genome of the mouse. And then asking the mouse, okay, without this gene, how do you respond to nicotine? What happens when I give you the opportunity to work for nicotine? What will happen to your liking of the drug? And what you can see here, it's fascinating work in that those animals that basically look like people have the hypomorph of the alpha 5 gene, here it's knocked out in the mouse. What you can see is that they love nicotine. So this is no nicotine at all, but as you increase the dose of nicotine here, what you're seeing is that these animals are taking more and more and more nicotine. There's no break. They continue to love the drug and continue to take a lot, just as a person with this variant will continue to smoke a lot of tobacco. In a normal mouse, you can see that at a higher doses, the nicotine actually becomes aversive. We believe it's because the alpha 5, the habenula, and other circuitry is kicking in, causing this aversion to high doses of nicotine, driving down the liking or the taking of nicotine. So you can see how this one gene variant is playing a very critical role in whether you like to smoke or not, or how much you might like to smoke or not. It's just one aspect. There's other aspects of tobacco addiction, how you metabolize nicotine. There are slow and fast metabolizers that's genetically linked as well. And fast metabolizers, as you might imagine, smoke more than slow metabolizers, and fast metabolizers like nicotine more than slow metabolizers because it becomes toxic. Remember, nicotine was used as an insecticide for many, many years, so you're actually taking in a toxin when you smoke. It just happens to be rewarding for a lot of people. I'll get back to that a little bit later in my talk. The other thing that's happening, of course, it's more complicated, right, than just genetics, because there's a whole area of epigenomics where you can modify gene expression without modifying the sequence of DNA. There are histones that surround the DNA that can be modified in ways that turn genes on and off, and we know drugs of abuse can impact this epigenome as well, and the HIV infection, which is a major area of research for NIDA. The HIV infection modifies the epigenome as well. So it's getting even more complex when you think about the genetic basis of drug addiction. But as I mentioned, it's not just genes. Development plays a huge role in thinking about vulnerability to addiction. And this is a slide from the NESARC study done at NIAAA showing DSM-3 at the time, dependence for various substances of abuse, whether it's tobacco, marijuana, or alcohol. And what you can see is there's a critical period between adolescence, 15 to 18 years of age, where there's a rapid increase in the incidence of substance use disorder. And looking up in the upper right-hand corner, this is actually work done at UCLA by Elizabeth Stoll, showing that one possibility for this effect is that the frontal lobes of the brain, Dr. London would know this very well, the purple areas here, are underdeveloped in the adolescent relative to adult. So there's less executive function, less inhibitory control. These are the areas of the brain that process that information. And when that's inhibited and you have a fully mature motivational pathway, drug reward pathway, you can see how there's this imbalance between the ability to inhibit and this tendency to be impulsive and take risks and enjoy drugs of abuse. And this is based on this scenario. You can see how you can end up with a pattern like this in terms of incidence of substance use disorder early in adolescence. So I think a key message here is prevention, 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 but this is the age group to really target when you're thinking about effective prevention strategies, and also thinking about how you might enhance frontal cortical function, executive function, given an immature frontal system. How would you do that? That's quite a challenge, but something worth discussing. We can take that again into the animal model and look at how young adolescent, in this case rats, P27 is about a 17-year-old, analogous to a 17-year-old kid, maybe 15-year-old kid, right? I don't know, maybe a little younger. But anyway, what you're seeing on the right then is these rats were given the opportunity to, again, self-administer nicotine here with a component in tobacco actually called acetaldehyde. RJR knew this early on, that by combining nicotine with acetaldehyde, it's much more reinforcing, much more potent than nicotine alone or acetaldehyde alone. And you can see here these adolescent rats simply love to take this combination. The number of self-injections is quite high relative to either component alone. If you wait till the animals grow up and give them the opportunity to self-administer, they have no interest whatsoever. So again, showing the importance of that developmental stage in the reinforcing properties of, in this case, constituents of tobacco. And again, this is just a study to show that if you look at early gene expression in ARC, which is involved in synaptic plasticity, rewiring of the brain, and you give adolescent rats nicotine, red being overexpression of ARC and blue being less expression of ARC, you can see a huge increase in the amount of ARC. So restructuring of the brain when you give nicotine relative to the adult brain. So again, suggesting there's a critical period for vulnerability to the properties, in this case, of nicotine in terms of both the reinforcing effects and plasticity, how it rewires your brain early in development, or potentially could. But so the third leg of the stool, of course, is experience. You have to be exposed to the drug. Once you're exposed, there's a whole cadre of events that happen. If many of you can think back to your Psych 101 Pavlov's dog, I mean, it's very common that we talk about addiction as a pathological learning and memory disorder, has a lot of mechanisms related to classical conditioning where exposure to the environment, exposure to the drug, causes a memory so that when you go back in that environment, you start to crave that drug over and over and over again, to an extreme case where you become sort of a compulsive user of that drug. And what a lot of work at NIDA has been doing is sort of mapping onto what are the brain regions and the brain structures that are involved in this pathological learning and memory disorder, from the amygdala involved in emotional learning to the hippocampus involved in long-term memory, the nucleus accumbens dorsal striatum involved in habit formation or long-term learning. We've talked about the prefrontal cortex as how that can potentially put the brakes on some of these compulsive behaviors, but those areas of the brain are compromised with extensive drug use, so this area of the brain is taking over in some respects and driving that compulsive drug use. So that's how we think about it, and because we understand the neural circuitry, we're then able to think about medications for treating addiction, and we've been targeting medications that involve various aspects of addiction. So to reduce the saliency of the drug, to prevent the drive, this compulsive drug taking, to reduce the memory, and these take on the forms of vaccines to interfere with the reinforcing effects of the drug, methods to improve executive control, strengthening the prefrontal cortex, other medications that may enhance extinction learning to reduce craving of the drug, etc. So there's a lot of interest in understanding the targets and developing medications that target these specific systems and molecular mechanisms associated with drug addiction, but the ultimate goal is to go from an addicted brain to a normal, unaddicted brain by normalizing the systems based on with medication and behavioral therapy, because we also do an extensive amount of research to tap into the same mechanisms, but from a behavioral perspective, where it's cognitive behavioral therapy, relaxation, and other techniques that tap into the same mechanisms, but do it in a behavioral approach as well. And ultimately, it's likely that we'll need both medication and behavioral treatment to solve it. It's not one or the other. Both approaches will be needed to have an impact. As I mentioned early on, when you saw the NIDA budget, if you caught it, I went kind of fast, but about $300 million, a third of the NIDA budget, is earmarked by Congress to devote to HIV/AIDS, and that's really because of the intertwined epidemic between HIV/AIDS and particularly injection drug use. You can see here the cases of diagnosis, percent diagnosis for injection drug use, but that's really just the tip of the iceberg. That does account for a substantial, steady amount of AIDS cases. But even when you think about male-to-male sexual contact, heterosexual contact, it's likely that drugs are involved in terms of engaging in risky, unprotected sexual behavior. Alcohol is disinhibitory, other drugs disinhibitory. People are at times engaging in risky behavior under the influence of drugs, which also drives the epidemic as well. So when you think about it, NIDA's interests run across all these subpopulations. The other thing that's been happening and really relates to, again, health disparities, when the disease was first recognized early on, it really came out of the white men who have sex with men community in major urban areas. But what's happening over time, and this is from the CDC 2009, and it really hasn't changed, is it's now moving into African American men, Hispanic men, and African American women, where the epidemic has moved, and it's become more rural and more in southern states. So how we think about treating and preventing HIV has changed dramatically over the last decades. And that's something we have to keep in mind when we're thinking about the face of this now epidemic has really changed quite a bit. And NIDA has an extensive program on HIV/AIDS looking at a lot of our funding, a lot of our $300 million actually goes into health services research, from looking, as I talked about, the racial and ethnic disparities in testing and access to treatment, and integrating treatment with substance use treatment as well. A large effort has been placed on seek, test, and treat and retain, which I'll talk about a little bit in my next slide. And as I mentioned early on in my talk, unfortunately, many of the patients or the individuals we want to treat end up in the criminal justice system. So the question is, how can we integrate services within that environment to treat HIV and substance use disorder while people are in prison, so that it gives them a better health outcomes and better chance of survival once they leave that setting. And we do have a very strong program in basic science, from looking at neurocognitive disorders associated with HIV to understanding really the genetic and epigenetic changes that go on when an individual is infected with the virus, and how drugs of abuse impact on those genetic and epigenetic changes. So really a robust, really exciting program in HIV. But I want to just get back to seek, test, and treat, because as you know, there's no effective vaccine for HIV yet. NIH is putting a lot of money into research in this area, private industry as well. But work by Julio Monteiro at Vancouver in British Columbia, Canada, really tracked what was going on early on in the course of HIV, and showed once HAART therapy was made readily available, when people were able to be tested and then given HAART therapy to reduce viral load, you can see back here before treatment, new HIV diagnosis is very high. Number of people actually on HAART was very low. As it started to become more part of the public health armamentarium in the late 90s, early 2000, you can see as the number of people getting onto HAART therapy increased, the number of new cases continued to decrease. What was also happening at that time, it's very important to remember, our substance use population is a stigmatized population. They fly under the radar. They're not tracked. They're not served by the public health system very well. In Vancouver, British Columbia, they made an effort to get to that population, to seek, test, and retain. And once they were doing that, the other thing that was happening is new cases for injection drug use was dropping as well as overall there were fewer new cases of HIV. So what this is suggesting is that HIV treatment as prevention is really a good strategy. And this was borne out by HPTN 52, which also showed with discordant couples, if you treat the person who has HIV, the other individual is protected. This really is a great way, without a vaccine, to think about how to reduce community viral load and reduce viral transmission of HIV. The only problem with this approach, it practically and conceptually makes perfect sense, right? If you can get everybody into treatment, give them therapy, you have a great opportunity to reduce community viral load and reduce transmission. We have a problem with the infectious disease doctors, though. Here's a study that was just published showing infectious disease docs' willingness to give heart therapy to injection drug users, even when their CD4 count, which we know is almost full-blown AIDS at this point, even when their CD4 count is 200. And what you can see here, percent of providers that defer are even at this very low CD4 count, 50% would not treat a daily injection drug user. So we have a huge problem here. We know this treatment is effective. We know it works. The docs are not willing to provide. It's not clear why. When you ask questions about can NIDA use it here, from this study there was a pretty high consensus that yes, they could adhere. But it's not clear what's driving this bias or the stigmatization. And if we're ever going to adopt a seek, test, and treat model, we need to get a handle on what's going on at this level, at the ground level, where the rubber meets the road. So I think this is a very telling study of where basic science, the research NIDA supports, is not getting out to the people that can make a difference, in the treatment of HIV. So I thought I'd just end in giving you some of the trends in substance use and see where we are, at least for individual drugs, and give you a sense of where things are going with this public health problem. And I think the biggest concern that's been happening over the last, really now 4 or 5 years, is the tremendous increase now in use of marijuana among Americans. 107 million Americans have tried it. 2.6 million use it for the first time. And then, of course, the majority, 2.6 million recent marijuana initiates were younger than age 18. And that's becoming a real problem. Early use of marijuana, it's now exceeded, this is the Monitoring the Future study that NIDA conducts every year of high school students. These are 12th graders. You can see percentage use of marijuana was spiked in the mid-90s. We were doing pretty well in bringing it down. I'm sorry, this is tobacco. And now tobacco we're doing quite well. Marijuana, though, for the first time we have this crossover effect where there are more high school students smoking marijuana than tobacco. And the problem with this is the earlier you start taking marijuana, the more likely you are to become addicted. 1 in 7 of these individuals will likely go on to become marijuana dependent. That's another issue to have to deal with in treating yet another substance use disorder, dependence on marijuana. The other thing we know, the acute effects of marijuana, devastating to learning and memory. Many of the receptors involved in the marijuana, the cannabinoid receptors, are on the hippocampus, which I showed earlier, critical structure for learning and memory. It's not good to have your kids high when they're in high school, and this is a critical problem moving forward. So the question is, what can we do to improve our messaging and think about how we can drive this rate of marijuana use down in our adolescent population? The other serious problem is psychotherapeutics. I don't know if many of you have been hearing about opiate medications. For the first time ever, opiate medication overdose deaths now exceed traffic accident deaths in the United States. More deaths from opiate analgesic medications than heroin and cocaine combined. So a huge problem. These are drugs that are being prescribed by primary care physicians to treat pain, that are being diverted, that are being abused, overused, and in many cases people are overdosing. So a huge problem in terms of how we think about getting a handle on this particular epidemic. When we look at across racial and ethnic minority groups, this is in one case where it's equal opportunity. Every racial and ethnic minority group is pretty much equally affected by substance use disorder. For illicit drugs, it runs about 8% across all groups, with the possible exception of American Indian, Alaska Native, where the rates tend to run higher for all substance use disorders. So clearly this is a huge problem regardless of race or ethnicity. What the problem, though, becomes is, again, we talked about treatment for HIV, it's also treatment for substance use disorder. The first problem is we have 22.3 million Americans who need treatment. Only 3.9 million receive it, 17%. That's awfully, terribly, depressingly low, actually. So how do we do a better job of providing the treatment that people need? That's number one, and ACA may go a long way to doing that. The Parity Act that was passed in Congress a few years ago now with Patrick Kennedy may help in providing those services under insurance, which is a good thing. The problem, though, when we look at who is admitted to treatment facilities, this is TED's data showing, again, it's predominantly white, with fairly good representation African American, not so good American Indian, Alaska Native. The problem, though, when you look at it by age, this is really troubling to me. It's when you look at the white population. Remember I talked about addiction as a developmental disorder. The quicker we can get in to treat people, the better the outcome. And it's happening here in the white population. You can see a huge spike in admissions for it. It's not so good for the African American group, whether you're male or female here. And certainly for the other ethnic minority groups, it's really quite abysmal. So we have a lot of work to do here in thinking about how we reach out to the younger individuals who are addicted early, particularly from other racial and ethnic minority groups, to get them into treatment sooner so we can have better outcomes later in life. It unfortunately gets even worse for prescription opiate abuse. These are admission data for treatment of opiate addiction medications, and you can see it's predominantly white and very little across the spectrum. Real problem here when you're thinking about treating this disorder. So what do we need to do? I think, and this is something NIDA's been out in front of, because drug addiction has been a stigmatized disease, it's not well taught in medical school, it's not thought about by primary care physicians, and not their fault. The time is of the essence when you're in primary care, as probably many of you know. It's not on their radar. If they were to diagnose, they don't know what to do with these folks. The integration between primary care and substance abuse treatment is not there. So even if you identify, what do you do? So there's a few things we've been trying to do, and one is we've developed a clinical trial network that consists of 13 medical schools throughout the country, and CTN also partners with Kaiser and other HMO communities to think about how we can do real-world intervention, evidence-based research, clinical trials in primary care to think about how we can do a better job of screening, of treating in that setting. And CTN has been doing a lot of work in that area to think about and run trials to improve this integration of substance use care and treatment and screening into primary care. And just one study that was done a few years ago, but I think we can do better, is the notion of screening and brief intervention and referral to treatment. It's called, another acronym for government, it's SBIRT. And the idea here is, while the patient is in the waiting room, and this was actually done in a primary care setting, in the waiting room, there was peer counseling being done where those that were self-reported cocaine or heroin use and later verified through hair samples were given a 20-minute brief motivational interview, given information about referral to treatment outside the primary care, and they were followed up for 6 months. And not, this is still a relatively, it's although significant, a small result, but in the right direction. Those that went through the SBIRT relative to control individuals that just received the pamphlet, there was some modest reduction in opiate and cocaine use in this group, suggesting that we can improve on this technique and provide that relatively brief intervention even during the waiting room time. It's also being tested in emergency rooms, people coming in for drunk driving or accidents due to drug use. They're given this SBIRT approach to see if we can integrate these individuals into primary care and also into substance use treatment. So this has been a major focus for NIDA for several years now. to see if we can improve. The other thing is education of physicians, and this is a partnership with Medscape, where we've put up, for CME actually credit, a web-based program to teach primary care physicians how to recognize and how to manage substance abuse for opiate medications. This gives practical, hands-on, there's actual scenarios that are acted through in this web-based tutorial that give physicians practical methods for recognizing, confronting, not necessarily confronting, but how do you negotiate with your patient who you think may be abusing their opiate medications. And it's been very successful. We've had thousands of hits, not just from primary care docs, but from other healthcare professionals, from pharmacists to nurses and others. And I encourage you, if you're at all in this area of primary care or dealing with patients, to take a look at this tutorial. Quite valuable and quite good. So that's really all I have to say. And I think, again, when I started, I said the only remember one thing from my talk is that, we're here to help. NIDA, all NIH officials, as you know, are paid by your tax dollars, so we work for you. So if you don't take advantage of us, your fault, because you're paying my salary right now for speaking and the things I do daily at the NIH. And I'm always happy to talk to investigators, whether you're seasoned investigators like the people over here, or early career people. I really like the early career people because I really get the opportunity to watch your careers grow. I remember people, when I started 18 years ago, that were pre-doctoral fellows and now are full professors somewhere. Or Laura Odell comes to mind, actually, who's speaking later today. So I just want to thank you again for your attention, and I'm happy to answer any questions you may have. So thank you very much.
Can you also go to the next slide?
Well, the National Institutes of Health is really the premier research organization for the federal government. We fund biomedical and behavioral research to address critical problems in disease. And as acting deputy director of the National Institute on Drug Abuse, our major focus is on issues around substance use disorder, addiction, and how to prevent, treat, and really understanding addiction as a brain disease, what really happens when people take drugs compulsively, how they become addicted, what happens to the brain of people that become addicted. And from that knowledge, we hope to then develop better treatment and prevention interventions.
And how many years have you been in your career?
Well, I've been at the NIH for over 18 years, and NIDA the whole time at the National Institute on Drug Abuse. And I just found it a fascinating place to work. I've learned so much more than I've ever learned as a researcher, because the breadth of research that NIDA supports, from molecular and cellular neurobiology through health services, prevention, epidemiology, I'm really getting a better sense of the complexity of substance use disorder. So it's been a fascinating and rewarding career so far.
Right, and it was so impressive how you took those 18 years and tried to break it down in 45 minutes, and I felt like I got speed training myself.
Well, thank you.
What do you feel is one of your biggest challenges with your job?
The biggest challenge, there's many challenges. So first of all, we just don't yet completely understand the phenomenon of substance use disorder. As you heard from my talk, there's a complex neurobiology system that's involved, complex social behavioral interactions that are involved in drug addiction and drug abuse. And we're still really studying those fundamental processes that are involved in addiction. The other real problem, though, and I mentioned in my talk, is translating the science we have supported into real practical treatment and prevention that will help people. And that's sort of the next hurdle. It's how do we get this knowledge that we've been generating? NIDA is going to be turning 40 years old in 2014. And that 40 years of knowledge base, how do we get that to the practitioners, to prevention strategies, to policymakers, to really make a difference to those individuals afflicted with substance use disorder? That's really the challenge ahead.
And how has your experience been here with this facility, with the school?
Oh, this has been great. I think this is so important. I really value what Charles Drew University is doing in terms of training medical students, thinking about health disparities. As I mentioned in my talk, there's really not a disparity in terms of the racial and ethnic minority groups that are afflicted with substance use disorder. It's pretty much constant across all groups. Where the problem is how do we implement treatment? And I think Charles Drew, as training future medical doctors and helping them understand the importance of substance use in their practice, I think can go a long way to reducing some of these health disparities we're facing in terms of providing treatment to those most in need and those most underserved right now.
And let's jump way far into your future prior to you retiring, if you ever really retire.
I don't think I ever will.
Right, they never really do. What do you think your dream goal would be, your biggest achievement would be?
Yeah, so I think having the science that NIDA supports, NIDA is a grant-making organization. We fund major medical schools like Charles Drew and others and universities. My major goal is that the science we support will be useful and used, and that for the long term that drug addiction, substance use disorder will be treated like any other disease, like cancer, like heart disease, like diabetes as a chronic relapsing condition that needs care, that needs awareness from the primary care community, that this is in fact a disease that is treatable, that can be managed, but that the primary care community be aware of this and implement the best practices from the research that NIDA supports.
And do you have any advice or support for the current students?
I would say for students, this is an exciting area of research. I think it's a great opportunity to grow and become involved in substance abuse research, so to encourage anyone at all interested in this field to continue. Right now we're going through a rough patch in terms of budgets, but we've gone through rough patches before. We've always come out the other end stronger and better, and in some ways, with the reduction in budgets, we're making decisions, really critical decisions, and prioritizing our research, which is a good thing to see what we really need. But I think for young investigators is to stay with it, be persistent, don't give up. And as I said in my talk, it's really important that they reach out to NIH officials like me and others who can really help them navigate through the complex process of getting a grant, getting a research grant. It does happen, but you have to reach out to the right person at the NIH, and you have to be persistent, you have to be passionate about what you do. And if you have those ingredients, you'll succeed.
And anything else you would like to add?
I just want to thank Charles Drew for inviting me and Dr. Friedman for hosting this event. I think it was fantastic. I think it's fantastic. We have a whole afternoon to go, and I'm looking forward to the talks ahead. And I think it's a great annual meeting, and I hope you'll continue it in the future.
Thank you very much.
Thank you.